Welcome to the first seminar in the Soil, Root and Nutrient session. My name is Malcolm Bennett, and in this session I want to focus on uncovering the hidden half of crops using advanced root phenotyping pipelines. My presentation will draw on experiences and recent research as part of the European Plant Phenotyping Network 2020, European Research Council and a US RPRE funded programme. So, why is root phenotyping so important? Well, to date, breeding programs have primarily focused on shoot traits to select crop varieties. However, we all recognise the importance of root traits like depth, angle and branching for resource capture efficiency. For example, rooting depth aids securing water, particularly during drought. Root angle aids foraging for nutrients like phosphate, which accumulate in the topsoil. And root branching density aids capture of nutrients like nitrate. Nevertheless, root phenotyping is very difficult given the practical challenges of imaging roots in their natural soil environment. So in today's talk, I'm going to describe two topics relating to root phenotyping. The first relates to imaging roots grown in controlled environments using examples from the European Plant Phenotyping Network and a European Research Council award. And in the second half of the talk, I'm going to talk about field phenotyping using an RPRE funded programme. So, as an example of one of the simplest systems in which you can image roots, this slide illustrates a very simple paper pouch based approach in which you can capture early seedling traits. They're grown on germination paper for up to 2 weeks, and you can capture very simple 2D root systems architecture traits to look at seedling characteristics. A more complex approach which allows you to look at many more plants and also at later stages of development has been developed by the University Catholique Leuven in Belgium, and this uses essentially an aeroponic system in which you can grow plants for much longer during their life cycle and capture more complex root systems architecture traits. Now, these two examples I've just given you obviously are devoid of roots growing in soil. One of the simple solutions, if you want to study roots growing in soil, are to use rhizotrons. And Jülich has pioneered this approach, developing some very elegant engineered rhizotron-based systems in which you can examine roots systems architecture growing in a thin layer of soil using multiple rhizotrons which are imaged by a centralized imaging station as illustrated in this slide. At Nottingham, we have been involved most closely with using X-ray CT imaging and imaging large soil volumes in a more natural soil environment. So this work was originally funded through the European Research Council and the Wolfson Foundation, Royal Society of the UK, in which is funded this facility called the Hounsfield facility. And in this facility we have an X-ray imaging building, and attached is a greenhouse complex. This arrangement allows for samples grown in the greenhouse to be transferred using a robot into the imaging station. So we use robots to move samples in the greenhouse, and then we use a robotic arm to move the samples into the scanning chamber. And whilst in the scanning chamber, this is a large lead-lined room which allows the sample to be scanned, rotated, and then up to 8,000 segments, CT segments to be taken, and then the samples returned to the greenhouse using the reverse robotic process. Now, obviously this system allows you to capture a large number of X-ray CT images, but there's a lot more work to be done before an image of the plant is actually generated. This requires the use of a CT-based root phenotyping pipeline. And so after the X-ray CT image capture, we obviously need to recover the root material. We need to reconstruct that into a three-dimensional architecture. We need to quantify the root traits. We have to manage the data, and then we want to use this in clever ways, and this includes, for example, modeling and simulating the data to uncover traits. So this requires a large amount of computational input. So let's start following image capture. What does the data look like? Well, the data looks like this. This is an example of a two-dimensional slice, CT slice, and you can see the soil particles, and within that matrix you can actually see highlighted in blue a root. And this reveals it's almost like a needle in a haystack within the soil matrix. To actually identify roots, we use a variety of approaches, including tracking, and more recently we've started to adopt the use of deep learning based approaches in a recent paper by my colleagues Mike Pound and Tony Pridmore. In this, they actually use deep learning based approaches which have allowed unprecedented abilities to automatically reconstruct root systems architectures from CT volumes. We feel this is a game changer. This type of image analysis has allowed us to analyze the image stacks as fast as we can acquire them, and this really is a breakthrough on the part of our colleagues in computer science. This now means that we can scan an image in a realistic time scale of a matter of hours and capture root systems architectures. And just as an example of this, here we see how we've used time-lapse imaging from X-rays to reconstruct root systems architecture for wheat. So this shows how wheat grows during its life cycle. We've scanned it repeatedly. Remember, X-ray CT is non-invasive, and this allows you to take time points, and this allows us to build up a developmental time sequence of how roots grow and develop. And this reveals, for example, how the seminal roots are critical for the initial pathfinding during the early season, and during the later season we see this flush of tiller roots which are capturing the nitrogen and possibly late season water for grain filling. So this approach has really helped us understand the dynamic processes going on within the actual soil itself, and helps us to better understand the resource capture strategies that, for example, wheat adopts as a crop. This work was only possible through the efforts of a large team funded through the European Research Council, which involved not only plant scientists, but crop scientists, soil scientists, engineers, computer scientists, and mathematicians, as illustrated here. I should like to also point out and acknowledge I'd like to switch focus now to address field phenotyping and to discuss some work that we're doing as part of the DEEPer project. The DEEPer program comes from the ARPARI Roots program, in which we're attempting to develop root phenotyping approaches to identify deeper rooting varieties of maize. It's estimated that if we're able to grow maize roots half a metre deeper than currently, we could bury approximately 2/3 of the US transportation-induced carbon footprint. So the idea behind this program is to develop advanced approaches to look at, for example, the architectural traits of maize varieties to identify deeper rooting varieties, and also look at anatomical traits which can confer deeper rooting. A whole series of these approaches are integrated within the program. So for example, we're developing invasive approaches based on the shovelomics approach, which involves removing the top of the root system and inferring various root traits. We're also developing non-invasive approaches in which we can infer depth, for example, from elemental composition of the shoot tissues. I'd like to talk a little bit about one of the invasive approaches that we're proposing, which involves the use of what we term the root robot. The root robot features a tractor pulled facility which automates the shovelomics process. So this involves a large degree of clever engineering, which is led by Paul Heinemann's team at Penn State, in which they have developed ways to remove the stalks from and root systems from the soil and then wash and prepare for them to be imaged in associated imaging pods. This next slide illustrates how we're using how the chisel takes out the actual stalk and the rootstock, and then this goes through a whole process of washing, which then is subsequently the rootstocks are then placed in imaging loaders, and then they're imaged using a clever arc of cameras in which they're able to obtain some extraordinary three-dimensional root systems architecture traits. The image analysis approach has been pioneered by Alex Bush's group at Georgia Tech, and they use cameras to allow multi-angle imaging and reconstruction of the rootstocks. In this movie image, we can see how the roots, which are computed, the images which are computed compared to the actual original image, are of breathtaking resolution. So we can see how accurately we're able to capture the underlying traits. And if we actually compare the automated traits, the accuracy of capture the automated traits to, for example, manually captured traits for the different root characteristics, we can see a very, very high level of accuracy. So we're very excited about this approach. This offers to reduce the backbreaking work associated with the root phenotyping that's now currently done manually. This could be a tremendous asset to speeding up this process of root phenotyping, not just for maize but for other crops, which this system could be adapted to analyze. And so the associated pipeline really helps us capture root systems analysis, and this we're using this to study, for example, traits which are associated with nitrogen capture as well as rooting depth. This is just one of the several innovative approaches which are being generated through the deeper program, which is led by Jonathan Lynch from Penn State, and involves 4 different institutions: Wisconsin, Georgia, and also University of Nottingham. Thank you very much for your attention. I hope you've enjoyed this brief presentation, and this illustrates the levels we're going to actually uncover the hidden half of plants and the new innovations which are often interdisciplinary to actually address this challenge but very important goal of capturing and harnessing root traits in modern agriculture. Thank you very much.